The project started with discussions that we were having on the anaerobic digestion technology. This technology allows us to convert organic waste into methane, which is a gas, energy-rich gas, which can be fed into the grid and used as an energy source.
The working engine that actually generates the methane are these very sort of complex microbial communities. And the big picture here is to try and optimise these microbial communities to make them more efficient at converting waste into biomethane.
Anything that's organic, that's grown or produced in the UK has to be disposed of if it isn't food. And the best way of doing that is mimicking nature and trying to degrade these biomass materials into energy.
The first reported use of AD, anaerobic digestion, in the water industry was to digest sludge in the 20s and 30s. And since that time, in one way you could say that the technology has come a long way, but most of those changes have been about changing the reactor format. But what we're really engineering when we're engineering an anaerobic digester is we're engineering a microbial population. It's only really in the last few years become possible to measure that microbial population in such a way that we could come up with feasible strategies. And that's what we've been doing in the LoLa project, finding new ways to make microbial communities work for us effectively.
What has never been really studied is how the microbial communities are working. So this is the new science that we are injecting here to sort of unravel the complexity and understand better the microbial communities. Hence, the insights we gain from studying these communities could be more generally applicable.
Microbes are really the most important organisms on the planet. They're responsible for keeping us alive, for example. The whole flow of nutrients on the planet is driven by microbes. So there are fundamental questions. We want to ask what organisms are responsible for these different processes, and we also want to understand how interactions between organisms drives these crucial processes.
Each reactor will have hundreds, possibly 1,000 species, and each species has its own genome, and when you add that up all together, you'll find that an anaerobic reactor will have 20 times more genetic material than the human genome.
Because we have so many species in these communities, it is very difficult to create a narrative in our heads about which species interacts with whom and where does that lead. And here at Warwick, we are focusing on modeling these systems as well as developing minimized versions of them. So the mathematical models and subsequent computer simulations allows us to formalize these interactions and to study the sort of time dynamics of the resulting systems. We try to feed those models with experimental data that we gather either from complex communities or from these simplified communities that we experimentally built. Those simplified communities usually involve either a couple of species or up to 5 species, but we are trying to capture the key interactions that we know are happening in the more complex communities. Some species would break down the complex sugar like cellulose into simpler sugars. Then other species might convert those simpler sugars even into simpler molecules. And there will be some conversions of those simpler molecules by yet other species. And finally, actually, the key species in the system are known as methanogens, which can access very simple organic acids and convert them into methane. So even though the complex community has thousands of species, I can create perhaps a 3, 4, 5 species version of it that still captures the key interactions going in that complex system.
Traditionally, we found the microbes which are most effective at degrading the waste and producing the methane work best at sort of 30 to 45 degrees centigrade. Now that's okay if you're treating food waste, you're getting a lot of energy off, you can use that to heat up the reactor. But it's not very good if you're treating very large volumes of domestic wastewater. As soon as we try and run those kind of reactors at anything much below 10 degrees C, then the reactors start to fail. The bottom of Lake Geneva is always at 4 degrees centigrade. The Arctic tundra is never much above 4 degrees centigrade. And yet we know that anaerobic processes take place there. If we get some of the bacteria which have adapted to those processes and get them to work in our reactor, then we might have a system which will work at the low temperatures that we want to get sort of energy positive wastewater treatment in the UK in the winter.
When we looked at all those different microbial communities, we realized that they produce methane at different levels. And that gave us the idea What would happen if we mix different communities that have a different level of gas production? Will they produce as much methane as the best individual component, maybe even more? There's some kind of a synergistic interaction, or are they going to fight with each other, and therefore the gas production of the mix is going to be worse than the average of its components? In order to do so, we designed an experiment when we took 10 different communities and we mixed them together, and the mix of the communities performed exactly the same as the best individual component of those communities, and it had a very, very similar composition as well in terms of what species are inside. So basically the strongest community does some sort of microbial nepotism, so its members establish itself in a new community and then allow their companion bacteria to also thrive and fill the same niches. Therefore, the gas production is very similar.
When we had so many different seeds from different parts of Svalbard and different parts of the settlement of Lake Geneva, we didn't know what to do, so we mixed them all together, and then fortunately Angus and his colleagues showed that that was actually the very best, most efficient thing we could have done.
The key point is that, of course, if you already know what the best performing community is, why would you bother to mix it? Well, actually, you don't know what the best performing community is until you actually set up the reactor, and it takes a long time to set up the reactor, and to test all these different communities in isolation you'd need many, many different reactors. So sometimes what you see in the lab on a small scale doesn't necessarily scale up to a large-scale reactor.
We're the process engineering people, so we are looking at reactors that will be quite large eventually, at 500, 1,000 cubic meters. What we want to understand is how these cultures survive over the long term in continuous flow systems, real systems basically, as opposed to individual little bottles or test tubes, etc.
The interactions between key species in the community are very much influenced by energetic aspects of the system. And we've been finding interesting consequences of that in terms of how much diversity we can expect in these communities. So it might be that one reason that there are hundreds and thousands of species in the system is that under those limited energy conditions, different species can coexist more readily than you would expect.
Our reactors now have membranes, so essentially you have a mass of organic material and bacteria. So this membrane filters out everything but basically liquid. So all the bacteria in the reactor are basically trapped there, and hence we have a chance of modifying the population. Can we get better populations to make more methane in less time?
We've been interested in anaerobic systems in Newcastle since the 1950s and even a little bit before. But right now, one of our big foci is how we can get anaerobic systems to treat domestic wastewater at low temperature. We believe that would be revolutionary for the industry, saving the UK populace millions and millions and millions of pounds.
I think one of the areas that is particularly interested me really is actually in general what is the consequences of the mixing of microbial communities together. Now again, this may sound trivial, but actually unlike most organisms, microbes are constantly being mixed up. But it's not just individual species, it's these whole microbial communities moving together. So one of the key questions to address really is do these communities act as semi-cohesive units? And if so, how? And this is really fundamental.
The project has been making significant scientific impact. We have been publishing high-quality papers that are describing new results. And I think it's also making industrial impact now, especially in the UK. We've been informing all of the anaerobic digestion plants and reactors of our science, of our results, and also getting their input in terms of what problems they're facing and what areas they'd like to see addressed. Those interactions actually led to a side project where we are now sampling many reactors in the UK in a weekly basis and recording their performance, but also recording their microbial communities in terms of composition. And this is now generating data for over a year, which is made publicly available, where people can see both the microbial community compositions and the reactor performances. But it has been also making impact in terms of generating more interest into microbial communities as well as anaerobic digestion. One thing we have organized is a 3-month residential meeting at the Isaac Newton Institute at Cambridge, which was attended by engineers, mathematicians, biologists, physicists, and has sparked many new collaborations. We have also created smaller interactions with colleagues in Netherlands, in France, in America, working on microbial communities and on anaerobic digestion. We like to think that this will facilitate new research in this area, as well as perhaps new policies and new economic forecasting, because you can then see how reactors are performing over longer timescales.
I think we're just at the beginning of engineering open microbial systems. There are 4.5 billion years worth of evolution that we can exploit to create solutions, not just in wastewater treatment, but in health, in agriculture, in veterinary medicine. We have only just started to realise what's out there and how the systems work. But when they do, we'll be able to ensure that when babies are born, they don't have ulcerative colitis. We'll be able to ensure that when we start up a reactor, it's going to remove pollutants we want it to remove. We're going to be able to get benefits from waste and things we currently throw away. I think they'll be intrinsic to the sort of low carbon, sustainable environment that we think we're going to have in the 21st and the 22nd century.